Good morning. I'd like to thank the organizing committee for inviting me to attend this incredible meeting, and especially the Chief Secretary, Ying-Yi Sun, and my friend and colleague, Professor Wan-Min Yang. It's been nearly a decade since the last time I was in China, and I really wish I could have been there in person. But such are the times we're living in. I'll start with a 10,000-foot view of the genomic era, past, present, and future, and then say a little bit about what we can expect in the coming decade. And I believe the coming decade will see transformative opportunities that are even greater than the past. And then I'll go on to discuss what I think is one of the great challenges facing the planet, certainly among the three most important in my view. And that is the contribution that genomics can make to addressing a rapidly changing climate and what genomics can do to help mitigate it and to adapt. The outline in the third slide is a reminder of just how transformative the genomic age has been. There's been a cultural shift in biomedical science from descriptive to information-based and predictive, routine use in drug discovery, substantially shortened pipeline, hundreds of actionable diagnostic tests, a revolution in the evolutionary sciences, a major impact in infectious disease epidemiology, a deep understanding of healthy and perturbed cells, important progress in social justice, and so on and so on. It goes on and on and on. Future advances, I believe, will dwarf those of the past. But what might those be? Well, prediction is always difficult, especially if it's about the future. I've tried this in the past, and needless to say, I'm not always correct. The next slide is a fragment from a talk I gave in 2011, at which I decided to go out on a limb and speculate a bit about what things would be like in the 2020s. The first, and this is something my friend and colleague Mark Batinsky was talking about even in the 1980s, is a more predictive and personalized medicine. And in that, I believe we've been reasonably successful. For example, we've been able to predict drug hypersensitivity, such as the relationship between abavakir and the HLA-B allele. Hundreds of actionable gene-drug associations identified by pharmacogenomic research now routinely guide treatment and dosing decision. With respect to a shortened timeline for drug development, that was already beginning to happen, so there was nothing new there. For the next one on the list, the way of new prognostics, there have been some, but not very much, so that really hasn't happened. The next item on the list, modest progress in treatment of complex disorders. I'll say more about that in a minute. There has been some progress, but some important progress was really not related to genomics. An integrated database for collective human genome and its environmentally modulated phenotypes, that really hasn't happened, at least not to the extent or to the way in which I had originally envisaged it. Major progress in infectious disease epidemiology, that's certainly been true. But what is the most difficult challenge? I don't think our most difficult challenge in the coming years is going to be scientific. I think the science is going to happen. The most difficult challenge we face, in my opinion, is catalyzing the diffusion of medical breakthroughs to the world's population. It's providing access to those who don't have it. We're now on slide 6. In 1986, Renato Delbecco published a paper in Science in which he called for a human genome project in order to speed research in cancer. And it has turned out that one of the great contributions of genomics has been a tremendous increase in our understanding of the cancer cell. But with respect to cancer mortality, the story is a little bit different, and this slide begins to show it. What is shown here is dramatic, but it's somewhat misleading because it doesn't correct for the world increase in population. The next slide does, and it shows that the increase in population, when subtracted out, has an enormous difference, but we're still left with a substantial increase in mortality during the last 30 years. The lowest curve is age-adjusted. The elderly have been downweighted. That's where most of the cancers occur. And the younger population has benefited substantially. And the reason, a good part of the reason, is that there's been a reduction in lung cancer and there's been a reduction in stomach cancer. It turns out neither of those really is tightly associated with progress in genomics. The lung cancer is the result of smoking, not smoking, and the stomach cancer is the result of having found the causative agent, which is H. pylori, which can be treated. And the same, in fact, is true of RCC, of renal cell carcinoma. There's been an association of H. pylori with that also. So we're moving forward in cancer slowly if you take out the aged population, but of course there is an aged population. So there is a problem, and we need to, we've made enormous progress in understanding the cancer cell, but translating that into a reduction in deaths has been very, very difficult. Slide 8 reminds us of a problem that I think is even more serious, and that is an inverse correlation between the wealth of a population and mortality. Wealthy populations tend to have longer 5-year survival times than low-income populations. Before going on to the next slide, it's helpful to provide some numbers for genotype-phenotype relations for a multi-gene disease. To be specific, if each gene has 3 or 4 major alleles, and if a disorder conservatively involved 4 genes, that would mean 64 to 256 phenotypes. And the number of phenotypes increases combinatorially with the number of genes. With 5 genes, the number of phenotypes could exceed 1,000. So establishing genotype-phenotype relations requires an enormous amount of data. And that brings us to the next slide. Taking the next step in understanding complex disorders would, I believe, be greatly facilitated by the availability of easily navigable public data warehouses with computational and modeling tools for integration, analysis, and conversion of data to knowledge. And I also believe if one really wants to understand most complex disorders, that database needs to go beyond genetics and model and include social, environmental, and genetic systems in order to understand both normal and pathological states. So I see progress here as being tied into a large project, massive data collection and analysis project. And analysis, I mean not just simple statistical tools, but modeling methods as well. And of course, this will also provide the data necessary for effective deep learning algorithms. And it's an area in which countries with large populations have a unique opportunity. Slide 10 reviews the social challenges. Most of the world's population does not have access to genomic progress, even in the world's richest country. It took 2 decades and hundreds of millions of dollars in the United States before any federal legislation was passed on privacy protection, and of course we're going to have greater issues in the future, much greater issues, and regulatory impediments, and we see this in all kinds of ways, and I'll be getting back to it when I discuss agrogenomics. GMO legislation has been a real stumbling block there. With respect to more specific predictions of what can be expected, NHGRI, the National Institute of Human Genome Research, has taken an initiative and called for bold predictions for human genomics by 2030. And this is slide 11, and this is what they have. And I think a lot of this would be great if it happens, and I think there's no reason why a lot of it won't happen. Generating and analyzing a complete human genome sequence will be routine from any laboratory. That's quite likely. The biological functions of every human gene will be known. Well, at least most of them will be, or many of them will be. Regular use of genomic information will have transitioned from boutique to mainstream in all clinical settings, making genomic testing as routine as complete blood counts. I won't go through the entire list except to note what's missing, and one of the things that's strikingly missing is any reference to complex diseases. Nothing about cancer, nothing about heart disease, nothing about the many, many neurological disorders. What also is missing is any reference to something that we're in the midst of right now, and that is infectious disease and pandemics. One of the most striking events that we've all witnessed during this last pandemic was the apparently rapid emergence of vaccines. It surprised a lot of experts in the field. Well, that emergence didn't happen overnight. Someone once told Louis Pasteur that he thought he was lucky, and Pasteur responded that chance favors the prepared mind. In fact, that's not only true of people, it's true in general of science. What happened now with the rapid appearance of RNA vaccine didn't just happen because there were developments in 3 months. There have been years of work that led up to that. So preparation is very, very important, and that's something I think that's going to need to be addressed because pandemics aren't going to go away, and we need rapid response to them all. We have rapid ability to make vaccines, to manufacturing them, but it takes facilities. So that type of preparation needs to be made. And that includes a better understanding of what organisms are likely to jump from animals to humans and how long it's likely to take them to make the jump. Okay, now we'll go on to another type of disease and another complex system, and that is the condition that's currently facing the planet. The planet has a fever, and I think genomics can help to bring it down. So how to respond to climate change requires understanding and manipulating the tightly coupled Ocean-Atmosphere-Terrestrial system. The change in climate, as everyone knows, is being driven by anthropogenic emission of trace gases, of greenhouse gases, the most important of which are carbon dioxide, methane, and nitrous oxide. And that accounts for 95% of the problem. And of those three, carbon dioxide is easily the most important. The current atmospheric concentration of CO2 is about 415 parts per million. Before the Industrial Revolution, or sometime between 1850 and 1900, the average atmospheric concentration was roughly around 275 parts per million. So there's been an increase in the past 150 years of so of approximately 140 parts per million excess CO2 in the atmosphere. That's about 1.1 trillion tons of excess carbon dioxide. The next slide, slide 13, illustrates the problem. If we stop emitting CO2 immediately, right now, today, what happens to that excess 140 parts per million CO2 in the atmosphere? The answer is that in the next 3 to 4 decades, it'll be reduced about 50%. And after 500 years, there'll still be more than 20% of that excess CO2 remaining in the atmosphere. Well, I think if we regard climate change as a land management problem, it can do a lot. The connection between land management and the composition of the atmosphere has been, of course, known for a very, very long time. And for several decades, people have been talking about modulating the climate by managing land better. About 5 years ago, there was a feature article in the New York Times Sunday magazine on that, how we can solve the climate problem with better land management. And what was striking about that article is there wasn't a word about the use of biotechnology. People just weren't thinking about it at the time. I wrote a letter to the Times, which was published shortly thereafter, suggesting that we're going to begin hearing a lot about this in the future. And then about a year after that, I organized a conference. But between that letter and the conference, I did some calculations, which were published in Biology Direct, just to illustrate roughly what can be expected if we were able to engineer trees. And some of the results of those calculations are shown on the next slide. We're currently releasing about 12 billion tons of carbon and carbon dioxide annually into the atmosphere. Under normal conditions, without the 12 billion tons emitted by humans, each year approximately 130 billion tons of atmospheric carbon are absorbed and then returned by terrestrial life during the annual cycle of growth and decay. The idea is to interfere with the return step, and there are many ways to do it. The red line shows the increase if CO2 emission is brought to zero uniformly over the next 80 years. This is what happens by changing, reducing CO2 only by regulation. There's no genetic engineering involved in this. It's just we have a goal of bringing it to zero over an 80-year time period, and this is what happens to the CO2. The blue line shows what happens if 10% of all trees, in addition to the regulation, 10% of all trees are engineered so that they return only 10% of the carbon they absorb. And you can see there'd be a huge impact. We'd be down to pre-industrial levels before the end of the century. Now, just to point out that it's important to regulate as well as to engineer, the diamonds show what happens if we just leave things alone at 12 billion tons a year released and just use engineered trees. And you can see the drop is very slight. It goes from about 850 billion tons to about 833 billion tons. And finally, the black line shows the effect of a delay in mitigation by 20 years. And you can see by the end of the century, there's not much of an impact, a little bit of an impact, but it takes a lot longer. So not delaying, if we're going to mitigate, not delaying, starting right away is very, very important. Okay, now I'll say a few words about some specifics in engineering, what it can be done, very briefly. The changing climate raises major problems in food security. That is, as the climate changes, certain plants, certain crops will not grow as well, and they're subject to drought, and they're subject to storms and everything else. So one important application area is to make drought-resistant crops. Julie Gray's lab at the University of Sheffield has been focusing on rice cultivars. Cultivating rice is very water intensive. What they're doing is optimizing the density of stomata on rice cells. These stomata are these pores in the surface that regulate the intake, the uptake of carbon dioxide, and the release of water vapor. So at the right density, the plant is more tolerant to stress such as drought and disease. And the same engineering methods can, of course, be applied to other plants, so this technique is very important in adapting to climate change. Another important way in which an impact can be made is by reducing the reliance on synthetic fertilizers. Fertilizers are currently produced by the Haber-Bosch process, which converts hydrogen and nitrogen to ammonia. And it could be one of the most important industrial chemical reactions ever developed. About 50% of the world's food production relies on Haber-Bosch ammonia fertilizer. But there are a number of problems. One is that it has a very large carbon footprint, and the other is that it's very poorly absorbed by plants. There are several potential bioengineering solutions by a few companies. One is by Pivot Bio, and that's a biological fertilizer. They've engineered microbes to produce nitrogen fertilizer from atmospheric nitrogen. You can think of this as being analogous to what plants do with atmospheric carbon. They convert it into an organic carbon, which is used by the plant. This process would use microbes to convert atmospheric nitrogen into nitrogen that can be utilized by plants. There's another, Anuvia plant nutrients, developed an organic matrix. This is a very interesting idea. That releases attached nutrients, avoiding runoff and the use of synthetic fertilizer. Nutrient runoff can cause algae growth and oxygen depletion in lakes and streams and harm fish and other forms of wildlife. And there's another approach, which is not bioengineering, it's not synthetic biology, but it's very, very interesting, and that's hydroponics. Vertical farms, indoor farms that don't rely on soil for growth. They have the advantage of requiring less land and less water. In fact, I've heard about a Shenzhen proposal for a skyscraper that would include a 51-story hydroponic farm. That would be utterly remarkable if it succeeded and well beyond anything that currently exists. Now I'd like to say a few words about photosynthesis. The fundamental fact is that photosynthesis is very inefficient, and the origin of that inefficiency is an enzyme called RuBisCO. As the diagram in slide 20 shows, RuBisCO catalyzes both oxygen and carbon dioxide. The carbon dioxide pathway is in fact leads to photosynthesis. The oxygenation pathway, called photorespiration, is wasteful. About 80% of all plants are subject to photorespiration. These include many common crops such as cassava, rice, wheat, and soybeans. These so-called C3 plants also include all trees. The other 20% of the plants have evolved adaptive mechanisms that ensure high concentrations of CO2 in the vicinity of RuBisCO. One is the C4 pathway that separates the reactions in space. The other is the crassulacean acid metabolism pathway that allows a plant to photosynthesize during the day, but only exchange gases at night. So that pathway separates the reactions temporally. Slide 21 lists a few ways in which photosynthetic efficiency might be increased. One is to concentrate carbon dioxide in the vicinity of the RuBisCO molecule. This basically plays on the two mechanisms that I just mentioned. Another is to engineer more efficient photosynthesis pathways. And then there are attempts to engineer more efficient RuBisCO molecules. There are difficulties with all these possibilities, which I won't go into, although my guess is that editing will succeed sometime in the next 4 to 7 years. But I'd like to say a few words about creating a transgenic using a more efficient RuBisCO, which can either be moved from one C3 plant to another, or by designing and synthesizing a new-to-nature molecule. One of the difficulties is that the activity of the molecule, its turnover rate, and its catalytic ratio tend to vary inversely with one another. It seems evolution has optimized the trade-off between activity and catalytic ratio, so that if you change one, the other one changes in the opposite direction. Now that's a statistical observation. It doesn't mean that in any particular case that's going to be true. There are, in fact, a number of instances in which one RuBisCO molecule has a higher activity and a higher ratio than another. The gain in ratio from a C3 to C3 transplant is, however, limited, although we don't know what that translates into in terms of crop yield. But we want to do more than just utilize natural C3 to C3 RuBisCO, because the gain from that would not be very large. We want to design new-to-nature RuBisCO with vastly improved characteristics, and we've developed and adapted machine learning algorithms to search large landscapes for such molecules. Okay, let me just quickly summarize. The genomic era has been and will continue to be transformative. In the coming decade, we can expect, among other things, increased emphasis on omic prognostics, especially in the complex disease arena, their integration with environmental and social data, infectious diseases, especially a deeper understanding of host pathogen interactions and monitoring, and a flourishing of agricultural genomics. The application to climate change, and more specifically a massive project, could result in a triple win: alleviation of the threat to food security, reduced atmospheric carbon, and the discovery of new classes of pharmaceuticals. The main obstacle to progress will not be scientific, but it will be cultural, including regulatory impediments to genetically modified plants and bacteria, and access to medical advances by low-income populations. Thank you.